Namaskar. Today we are here with another video tutorial, that is citric acid cycle, Krebs cycle. Krebs cycle is nothing, just opposite reactions of photosynthesis, Calvin cycle. That is, where carbon dioxides are attached from the atmosphere and converted into a food, that is carbohydrates, and that carbohydrates, that is carbon dioxide inserted in carbohydrate, are now released by oxidation. In a photosynthetic, that was a reduction of carbon dioxide, and in this case this is oxidation reaction. So it is a catabolism reaction here. Now we will discuss what is actually the cycle and what are actually the point you have to remember for competitive exams. So what are the point of concerns of this cycle? This is a 9-step cycle, and out of this 9 steps, 3 are irreversible steps. These 3 steps are irreversible steps, and rest steps are all reversible type. In this case, different type of reactions are there. Here is condensation reaction, here dehydration reaction, hydration reaction, dehydrogenase reaction, oxidative phosphorylation, and oxidative decarboxylation reaction, and the substrate level phosphorylation reaction. So all kind of reactions are there. Where are the different reactions? What are the different points of release of carbon dioxide? And what are the different points where GTP are produced, where FADH are produced? So we will discuss in details in every respect so that you can answer every question. So let's start the citric acid cycle, Krebs cycle, or tricarboxylic acid cycle. Why the name is tricarboxylic acid cycle? Because the first stable product in this case is a citrate, that is a citric acid, the first stable product, and it has 3 carboxylic group. First, you can, if we zoom it, this is a citrate and it has 3 carboxylic group, first, second and third. So why it is called tricarboxylic acid cycle. So let's the first stage. In the first step, what happened here is the irreversible reaction, and in this reaction, remember previously that the total reaction is occurring inside the mitochondrial matrix, not in the cytoplasm. Actually, the glycolysis reaction, that is when we eat the glucose is breakdown into it enters into the cell cytoplasm, and inside the cell cytoplasm, glucose is converted into pyruvic acid in a pathway that is called glycolysis. Glycolysis is a pathway in which the 6 carbon compound glucose is converted into pyruvic acid, that is the 3 carbon compound, and that pyruvic acid is entered inside the mitochondria. So inside the mitochondria, pyruvic acid is entered, and pyruvic acid is a 3 carbon compound. So from a single glucose, that is 6 carbon compound, is converted into 2 pyruvic acid, that is 3 carbon compound, and that pyruvic acid, after entering into the mitochondria, that undergo a decarboxylation reaction, that is pyruvate decarboxylase. Pyruvate dehydrogenase enzyme is responsible for this. For that from each pyruvic acid, that is 2 pyruvic acid is entered per 1 glucose, and from each pyruvic acid single carbon dioxide is released and 2 carbon compound is produced. This 2 carbon compound is called acetyl coenzyme A. So whatever the food we will take, whether it is carbohydrate, whether it is protein or fat, the fate is the same, that is they will all passing through the same cycle, and pyruvic acid will be produced and pyruvate will be entered inside the mitochondria, and then acetyl coenzyme A will be developed. And in case of fat, the acetyl coenzyme is directly entered inside the mitochondria, and that acetyl coenzyme is a 2 carbon compound here, CH3, C11O, and SCOA. This is a coenzyme A. This acetyl coenzyme is a 2 carbon compound, is a magical compound, is produced from pyruvic acid, and this 2 carbon compound and previously present oxaloacetate, that is a 4 carbon compound, this oxaloacetic acid that particular acid is too much important in biology. In existence of biology, there is oxaloacetic acid present, also present in C4 plants. You have seen that is a C4 plant, oxaloacetic acid is present. However, this is a 4 carbon compound. This 4 carbon compound is a first acceptor of acetyl coenzyme A with the help of enzyme citrate synthase, not citrate synthetase. Citrate synthase is very much important. Why the name synthase and synthetase? We will discuss later. It is the case of ATP production. In a reaction, when ATP is required, it is called tase, and when ATP are not produced or required, it's called ase. That is a condensation reaction. In this reaction, condensation means that is when oxaloacetate is joined with another small compound, two small compounds joined together. It's called condensation reaction. In this case, one water molecule is required. So this is the first step where condensation reaction takes place, and after this condensation, the first stable product is called citric acid or tricarboxylic acid. 3 carboxylic group, first, second and third carboxylic group is present. So why it's tricarboxylic acid, and the citrate will be immediately converted into cis-aconitic acid. Cis-aconic is the unstable product. Cis-aconic is it is actually intermediate product. It will be converted into citrate and cis-aconic, citrate and citrate. In that way, reversible reaction, and dehydration occurred in that stage. That is there. Dehydration, this water is released, and from cis-aconitate, from the 6 carbon compound cis-aconitate, the that is the isocitric acid will be developed. The isocitrate and cis-aconic, the basic differences are very less. Only OH and this OH group is present here, and in this case no OH group is present. So this OH group has come from hydrogen. This is another hydration reaction. So dehydration, hydration and condensation. So condensation, CDH, condensation, dehydration, hydration. This is a sequence: condensation, dehydration and hydration. Actually these questions are asked. These are very simple steps, but in a competitive exams lots of things you have to remember. So in that time you very rarely the students remember these steps. So this is a dehydration state, and isocitric acid is produced. All the 6 carbon compounds, 6 carbon, because oxaloacetic acid was 4 carbon compound and this is a 2 carbon compound. They combine together to form 6 carbon compound, and the 6 carbon compound is produced here. And the 6 carbon compound and isocitric acid. Now the first oxidative decarboxylation occurs. That oxidative decarboxylation means in this case oxidation reduction reaction occurs, as well as carbon dioxide is released. Which carbon dioxide will be released in this case? This first carbon dioxide is released. In this case it's a second carbon. This first carbon dioxide is released and produced from isocitrate to alpha ketoglutarate acid, and this isocitrate to alpha ketoglutarate, this is the first, this is the most important step, because too much phenomenon occur here. What happened here? The carbon dioxide is released, one molecule carbon dioxide released, as well as one molecule NADH are produced. That is reduced energy source. This reduced energy source will give ATP energy to us, to our cells in a terminal respiration. So this one is very important. We know that 1 NADH is equal to 2.5 ATP molecules, and 1 NADH is equal to 3 ATP molecule in a previous form. That is, previously it was assumed that the 1 NADH is equal to 3 ATP molecule, but in this case the 1 NADH is equal to 2.5 ATP, and 1 FADH we know that 1.5 ATP. So later we will discuss in a what are the stoichiometry in this case. So with the isocitrate to alpha ketoglutarate, this is a irreversible reaction, and this is a isocitrate dehydrogenase enzyme. So oxidation reduction occurs in this case. It is oxidized, isocitrate is oxidized, and NADH is reduced. So this one is very important step. This is a very important crucial oxidative decarboxylation station. And second step is another oxidative decarboxylation reaction. What happened with the alpha ketoglutarate? Alpha ketoglutarate acid 5 carbon compound. This 5 carbon compound will convert into 4 carbon compound with succinyl coenzyme A. Succinyl coenzyme A. With this enzyme, with the alpha ketoglutarate enzyme dehydrogenase. Alpha ketoglutarate dehydrogenase, and very important steps, because in this case another NADH is produced, and this is the oxidation reduction reaction. That is alpha ketoglutarate acid is converted as succinyl coenzyme A, and coenzyme is another again inserted and carbon dioxide is released. In this case, coenzyme is released in the first step, and this is the second, third, fourth and fifth step. Coenzyme is again entered. This coenzyme A is very important, and second carbon dioxide is released and produce the 4 carbon compound succinyl coenzyme A. This succinyl coenzyme A is converted into the succinic acid, and this step is a substrate level phosphorylation, and 1 GTP molecule is produced in this case. And this succinic acid is a 4 carbon compound. This coenzyme is again reduced, coenzyme is released, and this succinate will be converted into fumarate. These are not the isomer, none other than the isomers. So these are the dehydrogenation reaction, and these the succinate from succinate, when succinate to fumarate is produced, one FADH is produced. FADH2, this is another dehydrogenation reaction, that is a reduction. Oxidation reduction occur. FADH2, hydrogen molecules are only transferred from these 2H2 group is transferred from FAD to FADH2, and one double bond inserted between hydrogen group. This is from these and these. In between these 2 carbon molecules, one hydrogen bond is another double bond is formed, and 2 hydrogen molecules from one from here and one from here is transferred here to FAD to produce the FADH2, and the FADH2 will produce again in a terminal respiration the 1.5 ATP molecule. So in this case, it is very important that the fumarate is produced, and fumarate, the 4 carbon compound, is again with the help of water produce the malate, the malic acid. This is very important, and these two bond, if we carefully zoom it, that what happened here, the two double bond is there, not clear in this case. So from here the malate is produced, the malic acid, and this malic acid, from malate another dehydrogenation reaction occur where another NADH is produced, that is 2 hydrogen molecules are transferred and oxidation occur. In this case NAD is taking H plus ion and produce NADH2. So in the 3 places where NADH are produced, from malate to oxaloacetate, in this case first state isostarted alpha ketoglutarate to oxalosuccinate, and another step is malate to oxaloacetate, and FADH are produced in single case, that is succinate to fumarate. So these are the different places you have to answer. Question will be asked in that sense, that in which step carbon dioxide are released, in which step FADH are produced, in which step NADH are produced, in which step hydrogens H2 molecules are required, in which step H2 molecules are not required. So in this in that way, if we will discuss in a multi-directional way, and what are the steps of substrate level phosphorylation, which step the succinyl coenzyme to succinate in each case. So we can answer all the questions from this cycle. So let's start what are the single run. In a single run of acetic acid, what happened? Single run, in case of single cycle run of Krebs cycle, 1 acetyl coenzyme will be oxidized. 1 acetyl coenzyme. We know that from, if question will ask that from one glucose molecule, for one glucose molecule, how many times one Krebs cycle will run? So the answer will be 2 times, because 1 glucose means 2 pyruvic acid. 2 pyruvic acid means 2 acetyl coenzyme. We know that a single cycle can only oxidize single acetyl coenzyme. So 2 pyruvic acid for 2 acetyl coenzyme, 2 time run is necessary for this cycle. So what happened here? 2 oxidative decarboxylation reaction occur here. 2 oxidative decarboxylation. What is the isostarted alpha ketoglutarate and another alpha ketoglutarate to oxalosuccinic acid. 2 dehydrogenation reaction occur. One is for FADH production from succinyl coenzyme to succinyl to fumarate dehydrogenation reaction, another is malate to oxaloacetate. 2 hydration reaction, we know that the 2 hydration, the first hydration reaction is here. The first step, the first hydration reaction occurred. Second is this one, this hydration reaction is occurred. Another is actually the 3 hydration reaction is required, but it is a condensation reaction, so it's not a hydration reaction, but in this case the 2 hydration reaction is required for it. 1 dehydration reaction is required. What is the 1 dehydration reaction? 1 dehydration reaction, that is the first step here. After first step, in the second step, from citrate to cis-aconitate to dehydration reaction is required, and one substrate level phosphorylation to know that from succinyl coenzyme to succinic acid, there is the from succinyl coenzyme to succinic acid, the substrate level phosphorylation. You have to remember the substrate level phosphorylation present in also the in case of glycolysis. What happened in this case? In case of formation of 3 NADH in glycolysis, that is in production of 3 PGLD, then substrate level phosphorylation occurs. What is actually the substrate level phosphorylation? Substrate level phosphorylation, that is the production of ATP, that is or NADH, whatever maybe the phosphate group is transferred direct from substrate, not from any other source. So this is a substrate level phosphorylation. And another one is condensation reaction. You know that the first step is condensation reaction, where oxaloacetate to acetyl coenzyme is fused together. Oxaloacetate to acetyl coenzyme is fused together is the condensation reaction, and 6 reversible reaction is required and 3 irreversible reaction. What are the 3 irreversible reaction? 1 irreversible reaction is first is condensation reaction, and 2 decarboxylation. 2 oxidative decarboxylation reaction, that is the isociated to oxaloacetate and from oxaloacetate isociated to alpha ketoglutarate and alpha ketoglutarate to succinyl coenzyme A. So in a complete cycle, after complete cycle, 3 NADH is produced, 1 APDH and 1 GTP and 2 carbon dioxide produced. So for a single cycle this occur, and for in case of glucose, if we consider for a glucose, it will require it will be double, the value will be double, because for glucose 2 times the cycle will run 2 times. So it will be 6 and 2 FADH, 2 GTP and 4 carbon dioxide released. In a cycle, inside the cycle 2 carbon dioxide released, and before the cycle, that is from the formation of pyruvic acid to acetyl coenzyme, 1 carbon dioxide is released. So total 3 carbon dioxide released per cycle. One is outside cycle carbon dioxide released outside cycle during the formation of pyruvate to acetyl coenzyme. I am not saying here pyruvate to acetyl coenzyme, and 2 is inside the cycle. So this is a this is a phenomenon. What happened? The molecule of 1 molecule of glucose produce 2 molecule of pyruvic acid and 2 ATP and 2 NADH, and 2 pyruvic acid will be converted into 2 acetyl coenzyme by decarboxylation reaction. So for single pyruvic acid, 1 carbon dioxide and 1 NADH produced, but for 2 pyruvic acid is the 2 carbon dioxide and 2 NADH are produced. So this is a simplified form. You can remember in that way. Citrate, isociated, not cis-aconitate here. The citrate, isociated, alpha ketoglutarate sulfate. This is a practice way, and in this case amphibolic pathway is very important. That is, amphibolic means anabolic and catabolic both pathway are running together. So in this case, in citric acid cycle, it is assumed that is the catabolic pathway. Why? Because carbon dioxide are released in this cycle, but actually what happened in this case, both catabolic and anabolic cycles are produced, because the intermediate product alpha ketoglutarate and succinyl coenzyme, these are the intermediate products that can help the synthesis of amino acid and proteins. So amino acid production is synthesized from these amino acid production occur from these intermediate products. So why this is the anabolic pathway and catabolic pathway both running together, so it is known as amphibolic pathway. So intermediate products can help the production of different intermediate substances, so different protein synthesis or amino acids biosynthesis. So alpha ketoglutarate and succinyl coenzyme succinate, these are actually very important intermediates that help the production of different amino acids. So this why so why it is not called catabolic but also anabolic. So together it is called amphibolic pathway. Amphibolic pathway is amphibolic pathway. However, now the stoichiometry. Stoichiometry is very important, because we know that is a total 30 or 32 molecules of ATP are produced for a single molecule of glucose. So when we eat, we don't think that how much amount of ATP will produce for a burger or for a bread or a rice. One glucose molecule will give you 30 ATP molecule, one single glucose molecule. How? For the glucose, when we will take the glucose, it will transfer to the cytosol, that is cell cytoplasm, and inside the cell cytoplasm the glucose is converted into glucose 6-phosphate, and for this 1 ATP molecule is previously required, and the cell cytoplasm will give us a loan in the form of loan. Cell cytoplasm will give 2 ATP loan in the form of loan to the glycolysis system, so that the glycolytic system can run. So in a cell where previously no ATP is produced, no glycolysis will be started. So Previously ATP is required for starting the glycolysis. It's a very important thing, that is from glucose to glucose 6-phosphate and fructose 6-phosphate 1 fructose 1 6-phosphate. In both the cases 2 ATP are required in the form of loan. So you have to repayment the loan. Whenever you will take loan, you have to repay. So it is the glycolysis pathway also repayment the more loan. What happened in the 2 glycolysis? 2 glyceraldehyde 3-phosphate and then 2 1,3-bisphosphoglyceric acid. In this case 2 NADH are produced. This is the main problematic area, that is the 2 NADH. What happened? Why 2 NADH molecule is very problematic? In this cytosol these 2 NADH are produced, and these 2 NADH will enter into the mitochondrial matrix, and this entering pathway is different. That is, in some tissues this NADH produced inside the cytoplasm, that is from glycolysis, may be entered through one pathway, another pathway. We will discuss that in which pathway there is, and depending upon the entering pathway they will give 3 or 5 ATP molecules. So why the final product 30 to 32 molecules ATP will totally depends on the entering pathway, entering way of these 2 NADH from the cytosol to the mitochondria. This will be the major concern. And later, that is the 1,3-bisphosphoglycerate to 3-phosphoglycerate acid 2 ATP molecule produced. So in a glycolysis, that from glucose to pyruvic acid is the glycolytic pathway, and glycolytic pathway produce the 4 ATP and 2 NADH molecule, and these 4 ATP molecules, out of these 4 ATP molecules you have to repayment 2 ATP molecules in the form of loan. So loan repayment, it will be given 2 ATP molecule, and net gain is 2 ATP molecules. So in a glycolysis, if we will be asked that how much amount of ATP are produced, is the 2 answer, and 2 molecules and NADH are produced. Now the journey started from pyruvate to acetyl. Whenever pyruvic acid is produced, it will be entered into the mitochondria. Inside the mitochondria, from pyruvate to ultimately it will be total pyruvate will be oxidized, and all the NADH are produced inside the mitochondria, and this NADH will go to the terminal respiration. So in this way we know that in modern form FADH, one FADH molecule is equal to 1.5 ATP on an average, and one NADH in the form of modern form 2.5 ATP. So these all NADH produced in this case will produce 2.5 ATP. So in that case all okay, but why it is 30 or 32? This is a major concern. So as I mentioned that in case of liver, kidney and heart, these 2 NADH molecule from cytosol will be entered into the mitochondrial matrix through one specific pathway, one specific shuttle is called malate aspartate shuttle. If these 2 NADH will produce will enter into the mitochondria through the malate aspartate shuttle, then these 2 NADH will produce 2.5 or 3 ATP, 2.5 ATP molecule, kidney, heart and liver. But in case of skeletal muscle and brain, these 2 NADH molecule will produce will enter into the mitochondria in a different path. So why it will then give the 3 ATP molecule, sorry 1.5 ATP molecule per NADH. Per NADH now will produce 1.5 ATP in brain and skeletal muscle. So why the difference is 3 or 5? So if a question asks that if glucose is totally oxidized in a brain cell, how many molecules of ATP are produced? The answer will be 30. It will be, if we will ask that if one glucose molecule totally oxidized in a cell of liver, how many molecules of ATP are produced? It will be the 32. So this is the overall pathway of citric acid cycle you have to remember in a proper way. So thank you, keep watching.